Welcome back. Today we're going to conclude the look at the reproductive system by focusing mostly on the physiology of genetic inheritance. We're going to take a quick review of the anatomy just to bring you back to the idea of what's happening with the fertilized egg, and then we'll move on and look at some of the possibilities and some of the definitions that we use when we talk about genetic inheritance of various traits. Let's go to the first slide. And I'm going to run very quickly through these since you've already seen them, just to bring you a reminder of what we're looking at. Here is the ovum at the moment of fertilization. We have numerous sperm represented here, but this would number possibly in the millions. One has broken in, and through a calcium-mediated event, all other sperm will be kept out. Now, this is really a bit of an artist's fantasy, because what's going on here is the division already of the first cell into two, and that wouldn't happen with the sperm still out here. I thought it worth a moment looking at this. When cells divide, something called mitosis takes place, and this is the division where the chromosomes have already doubled their number and then divide so that each daughter cell will have the full complement of chromosomes. And this lineup of the chromosomes, here they show 4 on each side in their pairs, are going to be pulled apart at the spindle, and then this cell will divide down the middle so that you have the proper number of chromosomes on either side. What's interesting about this slide is we sometimes see this under the microscope, but the importance of it is not what you might think from this. In normal somatic cells, almost any tissue in the body, including the most actively dividing cells, for example, the GI tract, which turns over very fast, if you freeze or chemically fix the tissue at a moment in time, kill it right where it stands, slice it, and look at it under the microscope after it's been stained properly, you will almost never see what is here on the slide, the mitotic figure. Because normal, even rapidly dividing cells divide so infrequently that the chance that you have caught one in time and sliced that portion of the tissue that would show it is very, very small. It's so small that if we see this mitotic figure, we consider that evidence of cancer. Cancer cells are dividing so much faster that we see the mitoses frequently, and in fact, we count them to see how many there are, and the more there are, the more aggressive that cancer is. We'll talk more about that a few lectures from now. Moving on to the next slide, this is the first division, which is what we were seeing the beginning of in the last slide, and it goes on, and we have exponential growth as this ball of cells doubles with every division. Now, if you have an event right here in the first one, it is possible that when these two daughter cells divide, each now again with a full complement of chromosomes, and instead of separating just enough to make 4 cells, they completely separate and pull apart. That's how we get identical twins. If in one of these divisions, the two daughter cells pull apart from each other and no longer stay together, we have 2 organisms ready to go on and grow again, but they each have exactly the same chromosomes and the same DNA complement. The only difference they will have in the rest of their life will be those things in the environment, the nurture part, that will separate them. And the studies now done of separated twins are really quite remarkable for the similarities more than the differences. We then see here the egg moving on, dividing over this long period of approximately a week and implanting in the wall of the endometrium. And then as this is occurring, we start getting differentiation. Now, these cells, which were all the same a few days ago, begin to differentiate very early. They start getting chemical signals, which are still a great mystery to us, and one of the mysteries that we need to solve in this period of time when we're moving ahead in stem cell research. Because the cells of that first few divisions, up to the little ball, before differentiation, were called pluripotential or multipotential cells, or totipotential, which is the best term. Totipotential means that they can turn into any kind of tissue in the body. They have the ability to be directed down one path or another. The next stage that is occurring here is called determination. After we start getting some differentiation, meaning they just become different, you'll see here we have certain cells that are called ectoderm, meaning they're going to be on the outside of the body's tissues, and we have endoderm, which is going to be on the inside. They continue to differentiate further. They, in this slide, are now determined to be one or the other, and they will further become more determined. They'll start going down pathways. Some of the epithelial or ectodermal cells might become skin. They might become components of skin. They might become part of the gastrointestinal tract because that too is opened to the outside. That's epithelial. The endodermal cells might become other organs, but they can no longer go back. Once they've taken the fork in the road, they can never return. And this is very important when we get to our last lecture on cancer. So we have what is called now differentiation. And finally, when these cells get to the organ, specific organ tissue that they're going to develop into, they are now completely differentiated. And that's called terminal differentiation. This is all going on from information in the DNA, and yet all these cells start with the same DNA. Their DNA isn't changing, but they're going to get different signals. Let's move on now and look at some more slides. Here's the embryo now starting to look more and more like a real creature instead of a ball of cells, with always a disproportionately larger head. And I want you to just note the neural crest. These are the beginnings of the vertebral bodies, and the crest is open. This is where the nervous tissue of the spine is going to form. And it's open as if it's been filleted. It eventually has to close over. And there are deformities in which that does not occur. Finally, the baby here is implanted, looking very much like a baby with a disproportionately large head, as the fetus has at that point. And then finally, the most typical presentation, here we have something called the left occiput anterior presentation, in which the baby's head is slightly forward, which is anterior, and the back of the head pointing to the left. This is the most common for some reason. There are many other presentations, including a breech in which the baby's buttock and feet are at the bottom. Notice, too, how this baby has displaced everything in the abdomen. It's pushed everything out of the way to accommodate the baby inside the amniotic sac and the uterus. And here they don't show the uterus, but they just indicate it by dotted lines. And this has always been a nightmare for general surgeons such as I, who have to perhaps go in and take out an appendix here or a gallbladder there, and having two patients to deal with, one whom we don't want to disturb at all. And diseases of the abdomen during pregnancy are a great challenge, and all add quite a few gray hairs to you in the process. The last thing I want to show you is some of the things we can do for this baby. When the baby's in utero, we at that point are interested in possible genetic abnormalities, and some that we can fix and some that we can't fix. One very commonly performed test is called an amniocentesis. Centesis means the withdrawal of fluid, usually through a needle. In this case, the baby is in the amniotic sac still, and we are careful using ultrasound and palpation, feel, to see where the baby is and try to put the needle away from any vital organs. So we'll keep it away from the head and the eyes and the umbilical cord and the placenta. We can determine where those are, and we withdraw some fluid for examination. What we get is sloughed off cells, and we get some chemicals in there too, and we can measure those. We can look at the baby's DNA because he's going to have cells of his body floating in there. They can be extracted, and we can see if the baby has abnormal DNA. For example, 3 chromosome 21. And that way we'll know this is going to be a Down syndrome baby. There's another fairly common abnormality where the spinal cord fails to close completely. It's called a meningomyelocele, and the cord is left open. Oftentimes these babies are either paralyzed or have impaired neurologic function. Well, we can look for chemicals that are in that nervous system. One is called alpha-fetoprotein, which should stop being produced in the normal baby early on in pregnancy. Now, during development, if the neural crest doesn't close, that chemical, which should be in only very small amounts, ends up in large amounts in the fluid. And we know something may be wrong with the baby at that point. We also have a type of biopsy called a chorionic villi sampling, in which the attachment here can be sampled, putting a catheter through the vagina, up into the uterus, and taking a sample biopsy here. This can also be guided. Here's the ultrasound probe showing where the baby is. This can be done earlier. This is about 8 to 10 weeks, whereas the other one's about 14 to 16 weeks out of a 40-week pregnancy. Today, we have reason to do this more and more because there are some abnormalities which can be fixed. And the greatest extension of this technology is now intrauterine surgery. We can actually open the uterus, insert small instruments, and start trying to correct some of these defects. I don't know if any of you have seen the wonderful picture of an operation where uterine surgery is being performed. There's a small hole in the uterus, and out of that uterus has come a small hand, a miniature hand, and it's grabbed the finger of the surgeon. Makes every mother cry to see it. But it's where we're going in this. We're going to correct these defects sooner or later, earlier and earlier. Let's talk about genetics. That's now really the review of where we've gotten to at this point. The genetics involved here has to do mostly with what's going on in the DNA. We talk about chromosomes, and we'll continue to talk about chromosomes, but that's really the gross level of a view. The chromosome is really a long chain of coiled DNA that's brought together basically to a size we can see under the microscope. We want to remember always that we have either somatic cells, the body cells, or germ cells. The germ cells have half the number of chromosome as the somatic cells. So we call that body cell a diploid number. And we also want to remember that these are now fully differentiated cells that we're going to look at, but it doesn't matter where we take them. When we're counting chromosomes, it doesn't matter whether we take them from the stomach or from the brain or from anywhere else. We're still going to see the same chromosome count and the same DNA. The structure of DNA was elucidated by James Watson and Francis Crick in the early 1950s, and they presented us with what is the now famous double helix, two spiraling coils of chemicals that are joined together in the middle, primarily made up of a sugar-based backbone. And these chemicals are complementary. They have only 4 components in terms of different bases. They are called by their chemical names, but basically we talk about C, G, A, and T, and that's the chemical code. Now, each of the 2 of those 4 can only pair with each other, so that when they pair, they have a complementary strand on the other side. When they unzip and open up, we have actually 2 different chromosome chains, 2 different DNA chains, which are complements. They're mirror images. So when we lay down a new chain to replicate, we'll have exactly the same code now in the new helix that's put together, except we have 2 of them. This is the way DNA replicates, and this is the code that gives us all the information we have. Most of our DNA is gibberish. The vast majority of it has no meaning. It's just excess code. It's only probably as little as 10% that actually gives the information. Now, all this information is not transmitted into reality, into function, by the actual DNA itself. The DNA code is used in the making of proteins, and it's the protein or the gene product that performs the function that we need it to perform. So we can look at the code, or we can look at the gene product to see what that gene is supposed to do. And we can also tell by when genes are missing or damaged, what doesn't happen in that body or what happens that's abnormal, to also see what these specific genes do. The entire human genome has been elucidated now, and now it's a matter of finding out what the code means and how it works. Probably a bigger job than just figuring out the code itself. We need to talk about some terms. We have genetic differences. We have genetic diseases. We have the word genetic. This means anything that involves the DNA. It does not necessarily mean inherited, because things like cancer are caused by genetic damage to somatic cells that has nothing to do with inheritance. Fewer than 7% of human cancers are actually inherited. We have inherited changes. That means something that's been passed down to you in your DNA. And finally, we have an important word called congenital, which means with birth. It means that whatever we're talking about was present at birth. Now, this does not have to be genetic or inherited. You can have a baby that was born to a mother that was a cocaine or heroin addict. That baby has a congenital heroin addiction. The baby is born an addict, but may not have any genetic changes. They may, but they may not. They don't necessarily have to. Down syndrome, which we've talked about, is both congenital, because it shows up at birth, and it's also genetic and inherited. It's all three. We just have to keep in mind what we're talking about when we use those terms. We also have something called genotype and phenotype. The genotype is exactly the location and the character of the genes within the DNA. But the phenotype is how those genes are expressed. So the phenotype, for example, for brown eyes, is what we are seeing in the patient. The genotype will be the code that gives us the brown eyes. And sometimes those are a little bit different or hidden from us. We also have something called homologs, two chromosome pairs that are identical, that are the same number chromosome that we saw in our chart, are homologs. So we have homologs of each numbered chromosome as seen on this chart. If you look here, these are all the homologous chromosomes. And we also have something called alleles. If you look at the exact same location on two chromosomes, two homologs, we can pick location 17 on the short arm of chromosome 4. So we can look somewhere here. There'll be an identical place here carrying the same genetic information, or at least the same gene. One may be blue eye, one may be brown eye, but that's called an allele. It's the paired gene locations, or locus or loci as they're called. We also have to distinguish between dominant and recessive alleles. We have genes that express the phenotype more powerfully than another gene. One is called dominant, one is called recessive. And if you have two dominants, you may get one expressed trait. A dominant and a recessive will still give you that same expressed trait in most cases. I'm going to go through those very carefully in just a minute. And finally, two recessives together will give you another trait that may be totally different from the dominant. Most of our inherited diseases happen to be in recessive traits, but we have a couple that are not. For example, I've mentioned Huntington's disease, the neurologic disorder that happens to be an autosomal dominant gene. So just one of those inherited genes from a parent will give you the disease. So we also have incomplete dominance, which I'll talk to you about in a minute, and then which is when you can get a mixture of the traits and a moderating of the traits, as well as something else called homozygosity and heterozygosity. And that refers to when the gene complement, for example, back to number 4, if both chromosomes carry the same trait at that allelic spot, they are homozygous. If they carry different ones, dominant and recessive, then they are heterozygous, hetero meaning different. Let's take some examples and see which ones work to show us the different kinds of inheritance. Let's first talk about sex determination. We know we have X and Y chromosomes. The female ends up with a complement of 2 X chromosomes, and the male ends up with an X and a Y. Now, when these divide, of course, our germ cell is going to have one of each, and this square is called a Punnett square, and it is for working out these problems and testing probabilities. So if we have the sperm, the X sperm from the father, and we have an X sperm from the mother, we get a girl. If we have this X sperm going with this X sperm from the ovum, this is a girl too. But if we cross this one with this one, we get XY, that's a male, that's a male. We add them up, we get 2 XXs and 2 XYs. We have an equal number of male and female probability. Remember the trick about probabilities, that's all it is. You can have 7 daughters, and even though you had no other factors involved, when you shoot craps, every roll of the dice is an independent event. What happened before has no bearing on the next one. So your statistics are only good for 10,000 events, but not for any individual one. And these are just telling us probabilities. And that's why we have roughly an equal number of male and female births. There are other factors that affect it. It becomes very important in medicine, at least to me it was, when a patient would ask, what is my probability of dying in this operation? And the answer I always have to give is 100% or 0. Because for an individual, what happens to that individual is all or nothing. And it only is good, again, if you look at 10,000 patients, we can tell you risk, probability, insurance tables. But we can't tell you ever in medicine what's going to happen to you. So this is basic, simple transmission of the trait of female and male. The Y chromosome is kind of interesting. If we look at the Y chromosome again, it's this vestigial little shrimp of a structure that actually has no information on it. It is almost all gibberish except for the switch that turns on the differentiation toward maleness. Happens at 6 or 7 weeks. The genitalia, as we saw in both the female and the male, are the same, external and internal. Till about 7 weeks, we flip the switch here in the Y chromosome, and those that have the Y chromosome become males. Those that don't have it, that have 2 X's, are females. You heard me say it's dangerous to have more chromosomal material than you need. For example, trisomy 21 has a lot of abnormalities in the Down syndrome, mental retardation, heart problems, growth disturbances, and so forth. What happens then with the 2 X's in the females? That would be dangerous too. Shortly after this evolution of the embryo gets underway, we have something called X chromosome inactivation or degeneration. The X chromosome starts to shrivel up, the extra X chromosome in the female. One of the two randomly begins to disintegrate and go away, and it balls up into a chemically bizarre piece of material that stays in the nucleus and has different staining properties than the rest of the nucleus and shows up as a little dark ball in the corner of the nucleus called a Barr body and doesn't function anymore. Okay? Now, let's look at some more of these Punnett squares because they'll tell us a little bit about what's going on. If we take another very simple disease or abnormality called phenylketonuria. Patients with phenylketonuria have a genetic defect in which they can't metabolize an amino acid called phenylalanine to a very important one called tyrosine, and they end up with something called a phenylketone in their blood. These are very damaging things to have in your blood. They cause a whole bunch of symptoms, including some neurologic problems. And fortunately, if you drastically lower the phenylalanine in this patient's diet, they can get away with only minimum symptoms. That's why when you see sweeteners with aspartame, there's always a warning on the label not to take this if you have PKU, and because that happens to be one of the active ingredients in aspartame. Let's look at this Punnett square. The patients who have the normal gene are represented with a capital P. The abnormal gene for PKU is here. These are the small p, and these are the patients who cannot perform this metabolic task. Both these two patients are heterozygous for the trait in their homologous chromosome. So the mother and the father are both carriers. That means their dominant gene is able to carry out the function, so they don't need that other gene. And they don't have PKU. They are carriers that we would not know unless we did a genetic analysis. However, during the division of meiosis, where they separate these genes, they're now going to carry a lone gene for the disease. So if we look at the Punnett square, without belaboring it anymore, you can see what happens. We'll get a baby that is homozygous dominant, no disease. And then two babies, statistically, that are heterozygous with dominant and recessive disease gene, and a third that has both the disease genes, and this baby is heterozygous for the disease. We will then get one baby that is totally normal, two babies that are carriers but don't show the disease, and then one baby that has the disease. So you have a 3 out of 4 chances of being normal if both your parents are carriers. This is a very, very common mode of transmission of disease. You have the disease as a recessive, you have the dominant being normal, and you tend to get this heavily weighted 3-to-1 or 75% of the offspring don't have the disease, but you continue to produce 2 carriers in every generation of this. This is normal, dominant, recessive transmission. If we look at another kind where we have something called incomplete penetrance, this happens in a disease called sickle cell anemia. Sickle cell anemia is very interesting because it occurs primarily in blacks. It occurs in people who live around the equatorial belt. It is a disease in which, under conditions of low oxygen, that lovely red cell that was the flat biconcave hockey puck curls up, and it has a hemoglobin in it that is definitely defective. Now, these patients have been selected for along the equator. Evolution has selected them out in a positive way because it happens that the malaria parasite doesn't affect sickle cells as much as it affects normal cells. So there's been evolutionary pressure to propagate this disease because of another aspect of the insect world, meaning the propagation of malaria. In any case, it's not a good disease to have because the rest of your life is made miserable by what are called sickle cell crises. These patients are plagued with muscle pain, cramps, sometimes enlarged spleens, and a whole bunch of problems with their blood system. This is an example of incomplete penetrance. Here's what happens. We're going to represent the HB, which is hemoglobin, in these patients, and some of the hemoglobin is normal. That's A. Some of the hemoglobin is abnormal and has the sickle cell gene in it that causes this. We're going to look at 2 parents, both of which are carriers, and what happens is they're going to get the same distribution as we had before. But in this case, we're going to have a funny thing happen. We have one set of children, of offspring, that are going to get the capital A, the normal hemoglobin. They are going to be totally normal and not able to pass on the trait. We're going to have some, 2 of these, that are going to end up with A and S in theirs, and they're going to be carriers, and they're also going to have a mild form of it. It's called sickle trait as opposed to sickle disease. And they get a mild form. That's an incomplete penetrance of the gene. And then finally, one of the children is going to have sickle in both her hemoglobin genes. They're going to have the severe form of the disease, and they will be a carrier for the next generation. They're certainly going to give their child the trait because that's the only thing they can give. That's incomplete penetrance. There are lots of other bizarre ones. There's something called tasters. There are people that can taste a certain chemical and others that can't. And this is very much like the PKU. It's a very simple dominant or recessive gene. And then we have other ones that are sex-linked because they are traits or genes that are carried on the X chromosome. This brings us to an interesting problem. What happens here is, remember, the Y chromosome doesn't carry any information. And I'll get back to that as to why that's interesting too, but there is a red-green color blindness that's very common. It is a sex-linked trait. Here we have a normal male, because this male has the big C here and has a Y chromosome, and here the C and the small c, here's a heterozygous female. What do we get when we cross these? We have one normal female, XX, and has the normal color perception in the eye. We get another normal female who is a carrier but has normal color perception in the eye. We get a normal male because the Y chromosome doesn't matter, and then we get a colorblind male. This is going to definitely have a preponderance of male pattern color blindness. Something else going on in the Y chromosome that's awfully interesting, and by the way, this happens with all sex-linked traits. Anything on that X chromosome is going to have this kind of inheritance, and there are many others. The Y chromosome kind of gets the bad end of things when it comes to shuffling the deck. Look at all our other chromosomes. They're in pairs. They can line up next to each other, and they can exchange parts of limbs. Crossing over occurs before division. The two homologous limbs will cross, pinch off at the middle, and exchange genetic material. That's that shuffling I mentioned. So this evolutionarily is an excellent way to keep mixing the gene pool and always looking for the best combinations. And then when you deal the cards, you're going to naturally select in evolution for better and better organisms if it occurs before, the selection occurs before reproduction. The Y chromosome down here doesn't have anybody to exchange with. It doesn't have the same genes, and it doesn't have the same arms. There is no crossing over in the Y chromosome because there are never two Ys together. And also, it doesn't matter because there's no good information there anyway except the switch. But where it's become very useful lately is that my Y chromosome should be exactly identical to my father's and my brother's and my father's to his father's, and we should be able to take the DNA sequence in a Y chromosome and trace the origins straight back as far as we can find DNA material. And this has been done with numbers of tribes that have found themselves in bizarre places with bizarre social characteristics, making investigators wonder where they came from. And we can now start looking at the descendants, if we can get this material, as far back as we can go. It's going to be a very important arm of ethnic research. It's already been used for years in other lower forms of life to see what the evolutionary predecessor of certain animals were. Once we have this genetic material, and Jurassic Park wasn't really all that far afield. There are researchers who actually have the blood of Tyrannosaurus taken from Tyrannosaurus bones. And it's damaged DNA, but it's Tyrannosaurus DNA nonetheless. And though we're anywhere near making a new dinosaur, we certainly can get a lot of information about the offspring and the ancestors of lots of these animals. Let me talk to you about one more type of genetic inheritance, and this has to do with what's called polygenic inheritance. There are some traits, such as the color of your hair, color of your eyes, color of your skin, in which there are multiple genes involved. It's not just one. You aren't either one or the other. And in this case, we're going to show it as if there were 3 gene pairs involved. And if you got this generation of darkness, all of them being the dark skin, you'd have very, very dark skin. Here you'd have very, very light skin or hair, it doesn't matter. And then if you break these up into the 3s, you can get all these different combinations. Not worth detailing it, but you can see with this combination of genetic inheritance how we can go from the very, very light offspring all the way down to the darkest with all the gradations in the middle. We have all these different levels, and that's how we have such a multiplicity of complexions, of hair, of eye color, because we have polygenic many gene inheritance in these patients. There are a number of disease syndromes that are fairly common. If we went back to the female X chromosome, there is something called Turner syndrome, in which there is no second gene. There's just an X. We call that XO. This occurs in about 1 in 5,000 births, which isn't all that rare. And these are sterile females. They have no ovaries at all. They have short stature. They tend to have normal intelligence. There's another one called Klinefelter syndrome, which is only 1 in 500. That's fairly common. That's XXY. We have a duplication of the X chromosome at birth. We have slight mental retardation in these patients. They are sterile males with undeveloped testes, and they have breast enlargement because they have a predominance of the X, even though they're male, technically speaking. And we have something called XXX, which is a meta-female or super-female. These patients are mentally retarded. They are also infertile. They're underdeveloped. They tend not to have genital organs at all, of the sexual organs. So there's a whole bunch of combinations of things that go wrong, and unfortunately they do go wrong from time to time. We would like to know when we're going to have these children born. This is why we do amniocentesis or other biopsies. We then get into the moral dilemma of doing a procedure that may have consequences for the child or the mother if we're not going to do something about it. We don't want to do these just for the knowledge unless there's something we're going to do, either termination of pregnancy, surgery, chemical treatment, something that's going to change the events for either the baby or the parents. These procedures, for example, the amniocentesis does have an incidence of spontaneous abortion of about 0.5%. So 1 baby in 200 is going to abort at that stage after an amniocentesis, and you don't know if it's going to be your baby. You would hate to have this happen if you had a normal amniocentesis. The chorionic villus biopsy actually has a much higher one, maybe 1 or 2%. And that's when we start getting into significant complications. We have cases where the tissues of the chromosome don't separate, something called non-disjunction. We have several cancers that are caused by pieces of chromosomes staying with other pieces of chromosomes. So you end up with one pair that's short and one pair that's long. There's something called the Philadelphia chromosome that causes one of the leukemias that happens to be a non-disjunction, and that's a definite genetic event. I want to stop now from talking about these true genetic events, and I want to talk about some congenital events. And we don't always know whether these congenital events are genetic or not, but we see them frequently, and I think you want to know what they are and have heard the terms. These are going to be the basic heart abnormalities. And just take a look back at our first lecture. We talked about the evolution of the heart. Here we see the heart started as 2 tubes. This was the venous and the arterial, and they merged together. It began to form connections, became 1 tube, and then twisted around to become an atrium and the ventricle, and then finally the heart the way we know it. A 4-chambered heart separated into right and left. There are several abnormalities of this organ that is directly related to the failure of some of these components to actually go on the way they should. Let's go back to the slide. The first one here is called a patent ductus arteriosus. This is the one right here. There is a connection when the baby is an embryo and a fetus between the aorta and the pulmonary arteries. Blood flows, because we're not sending blood through the lungs of the baby for oxygenation, blood flows not only out to the body, but it flows back through the pulmonary arteries. So a little bit of blood may circulate through the arteries, but the rest of it's going to be divided up and mix with the arterial blood. What happens here is, if this doesn't close and the baby starts to breathe and his heart works as it should, we get increasing pulmonary artery pressure because this failure to close is now going through a working system. And we're putting arterial pressure, let's say 120 millimeters of mercury, into an area that's only used to having 30 millimeters of mercury. Much lower venous pressure. And so we get something called pulmonary hypertension. We also have a mixing that's going to overload both ventricles. Both sides of this heart are going to have to work harder. But this side, the left side, is going to have to pump out to the body and make up what's going through here. The right side is going to be pushing against this head of pressure, and it's not used to that. Very easy solution for this. We have the surgeon go into the newborn baby's chest and, in the old days, put a little tie on that, or today a stainless steel clip. This can sometimes be done laparoscopically through the ribs. Just go in, find the patent ductus arteriosus, put a clip on it, the operation's over, baby is cured. It's really quite a wonderful thing to be able to do for the baby. We have another defect called an interatrial septal defect. Remember that in the embryologic stage, we also had a hole between the two atria. We have the foramen ovale, and this closes because of the pressure changes. It's a flap valve that once the pressures are so different in the two sides of the heart with the first breath of the baby, this usually closes. In this case, it doesn't close, and a persistent hole remains open. It's usually very low flow, but blood will go, instead of all going into the ventricle, it will come through the patent foramen ovale, mixing blood from the right and the left, and again, increasing the load on the pulmonary circulation, because we've got too much blood flow coming in than should. It's got to receive the blood from the body, and now it's getting extra flow. And we repair this by either sewing the flap valve shut, or if there's no valve at all, going ahead and putting in a little patch, an artificial graft. That's done with open heart surgery. These babies have really benefited from the heart-lung machine. We have interventricular septal defects, same thing. In this case, the septum, which tends to grow up from the bottom and join the top of the ventricle, remains open. This is a much more serious defect because you have high flow going over to this side. It leads to the mixing of oxygenated blood between the right and the left, and it usually overloads the myocardium, the heart muscle, because, again, of high flow situations. And over here we have probably the most serious of them all, is called the tetralogy of Fallot. Dr. Fallot described this. Tetralogy means 4 defects. Here's what they are. First of all, the aorta, instead of being on the left side of the heart, overrides both sides. So it is a failure to separate the two sides. Second of all, the pulmonary valve, or the pulmonic valve, leaving the right side, is stenotic. It doesn't open very well. It's very tight. The third is an interventricular septal defect, which is right below. And finally, you get right ventricular hypertrophy and enlargement. This is what they used to call the blue babies. And the blue babies' problem was we were mixing a lot of deoxygenated blood with oxygenated blood and almost nothing going out to the lungs. Very poor circulation to the lungs to bring back good blood to the heart, and therefore the blood going out the aorta was literally blue, and the baby turned blue. This is a great surgical emergency, very high mortality. These babies needed to be operated on very quickly, depending on how severe each of the 4 things were. There are variations in how much they may or may not be overriding or stenotic and so forth. So some of the babies did better than the others, but basically this is one very sick baby. And finally, the other one that's very common is called coarctation of the aorta. The baby has a narrowing right here where the patent ductus used to be. And these babies have hypertension in their head and arms, and then hypotension in their lower limbs because they're not getting enough blood down there. It's a lot of stress on the heart, and it will affect growth and development of the baby, but this is very easy to either bypass or open up. Again, when you have proper cardiopulmonary bypass, all these operations are more simply done. Patent ductus arteriosus is the only one that doesn't require that at all, ever. Those are the things that can go wrong with our genetic inheritance as well as some of our congenital defects, and that brings us to the end of our exploration of inheritance and development.